In this video, we're going to be discussing reversible enzyme inhibitors. Now, these are going to be molecules that bind to enzymes reversibly, so via weak interactions, and somehow inhibit their function, not allow them to catalyze as well as they can. And the learning objectives are, I want you to be able to describe the experiments necessary to determine the type of enzyme inhibitor that you are examining. I want you to describe the effect of each type of inhibitor on an enzyme's Vmax and Km. And I want you to be able to look at enzyme kinetics data and identify whether that's data for which type of inhibitor. So first, let's say we have an enzyme inhibitor and we want to know what type of inhibitor there are. There's going to be three different types that we're going to be looking for. And the way that you do this is you run two enzyme kinetics experiment. First, you run one without the inhibitor to see what the actual Vmax and what the actual Km of an enzyme without the inhibitor is. So you do that the way we have described in the past, which means that you're going to take several different tubes, and in each tube there will be the exact same concentration of enzyme. That's ET here. And then with each tube you're going to have increasing concentrations of substrate. And for each one of these tubes you will calculate the initial velocity, the amount of product that is produced per minute at the beginning of that reaction. You will plot this out on an enzyme kinetics graph where you have the substrate concentration and is the x-axis and the initial velocity is the y-axis, and from that you calculate the Vmax and the Km of the enzyme. Now that's the actual Vmax and the actual Km because we do not have an inhibitor there. Then you do that same experiment all over again, okay, with the same enzyme concentration all the way across with increasing substrate concentrations, but this time to each one of the tubes you add a constant concentration of the inhibitor that you're interested in learning more about. And then you do the same thing. You figure out now in the presence of this inhibitor what is the initial velocity for each tube. You plot the initial velocity as a function of substrate concentration, and then you determine what's called the apparent Vmax and Km. And what you're going to see is that inhibitors change the Vmax, change the Km of that enzyme. That's why we call it the apparent Vmax, the apparent Km. It's not the inherent Km or Vmax of the enzyme. It's how the Vmax and Km are changed in the presence of the inhibitor. And so how that Vmax is changed and how that Km is changed will allow us to determine what type of inhibitor we're examining, that we're looking at. So there's three types of reversible enzyme inhibitors that we're going to discuss. And the first, and maybe the simplest, the easiest to understand, is called a competitive inhibitor or competitive inhibition. So I, E is our enzyme, S is the enzyme substrate, and I is the inhibitor. And the inhibitor, in a competitive inhibition, the inhibitor binds to the same place on the enzyme that the natural substrate does. Okay, it binds the active site of the enzyme, which means that the binding of the substrate or the inhibitor to that active site is mutually exclusive. If the inhibitor is bound there, the substrate can't also bind there. And so once the inhibitor is bound, substrate can't bind, and that enzyme can't catalyze a reaction. Now, there's a few interesting things here. First is because the inhibitor binds to the same place as the substrate, it binds to the active site, then if you knew the structure of the inhibitor, you can start to make some predictions about what the structure of the substrate, the natural substrate, might be. And in fact, biochemists often use this as a way to determine the structure of the substrate of the reaction. Now, the inhibitor binds the enzyme And the only way that we'll get that enzyme to be rid of the inhibitor is to saturate it with substrate. So if we added more and more and more substrate to that reaction, we would eventually be able to outcompete the inhibitor for binding to the enzyme. And because we would have to add more and more and more substrate to do that, we're going to end up increasing our Km. Remember, our Km is going to be the substrate concentration at which half of the enzymes are saturated. And so now we're going to have to add more substrate, and therefore we're going to increase the Km. The maximal velocity, however, will be the same. Okay, so let's take a look at that. Okay, here is some enzyme kinetics data that demonstrate that competitive inhibitors raise Km but don't change the Vmax. The black line represents the data without any inhibitor present, and the pink line represents an experiment done in the presence of a competitive inhibitor. This is a double reciprocal plot, so the y-axis is 1 over V, and the x-axis is 1 over S, which means that our y-intercept is 1 over Vmax, and notice it's the same. The x-intercept is negative 1 over Km, and notice that they are different. And in fact, you can see that the Km has increased with the presence of a competitive inhibitor. Now, the question is, by how much? Right? How potent, how strong is a competitive inhibitor? Well, that strength of the inhibitor is defined by this term alpha. That's called the apparent factor. It is the factor by which that Km is increased in the presence of the inhibitor, which means the higher the alpha, the more potent the inhibitor. I've got a question for you. What factors might go into determining the magnitude of alpha? Think about that for a second. Pause it. All right, so let's come back, and hopefully you've put some thought into this. What factors go into determining the magnitude of alpha, just how potent that enzyme is? Well, there are two main factors that go into it. First, the concentration of the inhibitor. The more inhibitor there is, the more potent it's going to be. And the second is right here, what's called Ki, and that's the equilibrium constant for the enzyme binding the inhibitor to become an enzyme inhibitor complex. And that constant, right, that dissociation constant is defined, or equilibrium constant is defined by this Ki. And notice it is the concentration of the free enzyme times the concentration of the free inhibitor divided by the concentration of the enzyme inhibitor complex. So if we think about that, the more effective the inhibitor, the blank Ki would be. So the more effective means the more we're leaning towards the concentration of the enzyme inhibitor, and so the smaller that Ki is going to be. So what we have then here is a situation where alpha is equal 1 plus the larger number divided by a smaller number, which means the higher alpha is, the more potent that enzyme is going to be. Now, many drugs are themselves enzyme inhibitors. In fact, that's the way most drugs work. You take them, they get into your cells, and they inhibit specific enzymes in specific biochemical pathways. Let's take a look at an example here. Ibuprofen is a drug that acts as a competitive inhibitor of two enzymes, COX-1 and COX-2. Now, these enzymes are involved in a biochemical pathway that leads to prostaglandins involved in platelet activation and kidney and liver function. And COX-2 is involved in a biochemical pathway that leads to prostaglandins that mediate inflammation and pain, which means when you take ibuprofen, you are inhibiting these two enzymes, inhibiting these two biochemical pathways, and therefore you are reducing inflammation, reducing pain, but at the same time, because this enzyme also inhibits COX-1, you're also, if you take too much of it, can alter your kidney or liver function, and you will reduce platelet activation, which means ibuprofen will lead to what's called blood thinning. And this is important because ibuprofen can be used, and aspirin also inhibits the same enzymes, can be used if you are susceptible to strokes, if you have a heart valve put in, you would be taking aspirin once a day, every day, to reduce platelet activation and minimize the chances of blood clotting when it's not supposed to. So, as I said, ibuprofen is specifically a competitive inhibitor, which means it binds to the same place that the natural substrate binds to. So here's COX-1, and here is arachidonic acid, the natural substrate, and there it is right in the active site. And if we take a look at ibuprofen and aspirin, same thing. They bind in that same site, inhibiting the ability of the natural substrate to get in there. Okay, so ibuprofen is a competitive inhibitor. Lots and lots of drugs are reversible enzyme inhibitors. Next, uncompetitive inhibition. In uncompetitive inhibition, the inhibitor doesn't bind the active site, it binds a completely different region on the enzyme. And it can only bind there when the enzyme is bound to substrate. And so what you get is this new complex, this enzyme substrate inhibitor complex. And when that inhibitor is bound to the enzyme substrate complex, then the enzyme cannot catalyze the reaction. So what this effectively does is that the concentration of enzyme substrate complex is reduced by the concentration of the enzyme substrate inhibitor complex. And what that means is that it's going to artificially reduce the Km. It's going to take less substrate to saturate the enzyme. And so, again, that is going to reduce the Km. But at the same time, since we are reducing the concentration of enzyme substrate, effective enzyme substrate concentration there's going to be less ES, and therefore our Vmax is also going to be reduced. So if we take a look and plot our data out after we do an enzyme kinetics experiment in the presence of inhibitor, this is what we'll see. Here in blue is no inhibitor. It has a specific actual Vmax. It has a specific actual Km. And as we add a uncompetitive inhibitor, notice that we are reducing the Vmax and we are reducing the Km. And the key here is we're reducing Vmax and Km equally, reducing them by the same factor. That's important, especially when we start to compare uncompetitive inhibitors with mixed inhibitors. So what defines the strength of an uncompetitive inhibitor? And that is another alpha called alpha prime. So the apparent Vmax is going to be equal to the actual Vmax divided by alpha prime. The apparent Km is going to be equal to the actual Km divided by alpha prime. What is alpha prime? Well, it's very similar to alpha. It's going to be 1 plus the concentration of the inhibitor divided by K prime I. K prime I is the equilibrium constant for that enzyme substrate complex binding to the inhibitor to make the enzyme substrate inhibitor complex. Okay, and so that is what alpha prime is, and that is the factor by which both Km and Vmax are reduced, thereby giving us data that looks like this, these parallel lines, because Km and Vmax are being reduced by the same factor. The last type of inhibition that we're going to look at is called mixed inhibition. And this type of inhibitor interacts with both the enzyme alone and the enzyme substrate complex. It is a mix of both competitive and uncompetitive inhibitors. And so what that means that it's going to decrease the Vmax for sure, but it may increase or decrease the Km. And the question is, it sort of depends upon its affinity. Okay, so that inhibitor will have an affinity for the enzyme alone, defined as Ki, and it will also have a separate affinity for the inhibitor to the enzyme substrate complex. That's called K'. And depending upon these two affinities will determine whether overall this inhibitor is going to increase or decrease Km. So here's some enzyme kinetics data that demonstrate that mixed inhibitors decrease the Vmax and change the Km. And again, this is double reciprocal plot, so this is 1 over initial velocity, this is 1 over substrate concentration. In blue is no inhibitor, and then in black is with increasing concentration of inhibitor. And what you'll notice is, sure enough, we see decreases in Vmax as we increase the inhibitor concentration. We see increases in Km as we see increases in the concentration of inhibitor. So mixed inhibitor always decreases Vmax, and they always change the Km. But depending upon the mixed inhibitor, sometimes the Km, the apparent Km, will increase, or sometimes the apparent Km may decrease. Now, the strength of mixed inhibitors is defined by both alpha and alpha prime. Alpha takes into account the inhibitor's affinity for just the enzyme, so Ki, alpha equals 1 plus the concentration of the inhibitor over Ki. And alpha prime takes into account the inhibitor's affinity to the enzyme substrate concentration. And so what we get here is that the apparent Km, the Km in the presence of the inhibitor, is equal to alpha times the Km divided by alpha prime. The apparent Vmax, the Vmax that you see in the presence of the inhibitor, is equal to the actual Vmax divided by alpha prime. Here is a nice table that you can get from your textbook that demonstrates how the 3 different inhibitors change the actual Vmax. So if we don't have any inhibitor, we have our actual Vmax, we have our actual Km. And then in a competitive inhibitor, the apparent Vmax is the same as the actual Vmax. Competitive inhibitors don't change the Vmax. However, competitive inhibitors change the Km by this alpha factor. They increase the Km by this alpha factor. Uncompetitive inhibitors take that actual Vmax, take that actual Km, and reduce them by the same factor, alpha prime. And finally, mixed inhibitors always reduce Vmax by alpha prime and change Km by a factor of both alpha and alpha prime. Okay? So make sure that you understand these and that you can derive what type of competitor inhibitor you're looking at by looking at the changes in Vmax and Km. So again, that brings us to our learning objectives. Make sure you can describe the experiments necessary to determine what type of inhibitor you're looking at. Make sure you can describe the effect of each type of inhibitor on an enzyme's Vmax and Km, and be sure to interpret enzyme kinetics graphs for each type of inhibitor.